Okay, and hello to you. I realize I'm the last thing between you and lunch. I'll try to be fast as usually. There are two things which I'll try to cover explicitly, and that's nonlinear functional connectivity and the higher order effective connectivity. And there's actually a lot of context, which I sort of made colorful for two reasons. One of them to sort of create some potential links for discussions in between you or in between the whole complex systems of this workshop. Thanks for having me. And secondly, because you might notice there might be some remnants of a previous skeleton of a psychedelic conference talk. So don't be disrupted by psychedelic references at maybe one or two of the slides. That doesn't work. I'll use this one instead. Doesn't want to disappear. So this is what I will not talk about, and that's modeling explicitly the dynamics of the brain by connecting some locations to a network through structural connectivity using diffusion imaging, and basically putting some nonlinear dynamics on it and simulating the thing. This is a great thing to do. This is the ultimate description we'd like to have of brain activity, to have the equations of motion. But I will not show, almost not show any equations of motion in this talk. I will do more of this type of game, and that's just observing people by a weird way of looking inside their brain instead of from outside. But we somehow believe that we can say something about about humans by looking at the brain or brain activity. But it's not the equations of motion, not too much. It's purely observing, and there are many methods of observing. I'm in a good position to be after speakers who said, especially the starting keynote, a lot about observing the brain. So I can be very fast in here. And I will just mention one particular thing, I'll just highlight. There has been a shift roughly 20 years ago. It's not a complete transition, but it's a shift maybe of the center of mass in neuroimaging after the appearance of spontaneous fluctuations actually having meaning in functional magnetic resonance imaging. So when fMRI was observed for the first, let's say, 10 years, it was predominantly used for mapping brain function through tasks. We give you a task, move fingers, and we find out that your motor cortex lights up. But actually, later on, partly by coincidence, scientists realized that if you observe just spontaneous activity during 10 minutes, or in this case, I think it's 5 minutes of purely resting state, do not do anything in particular, just lie in the scanner, try not to move too much, try not to fall asleep. It should be even simpler, the condition. There is spontaneous activation, not perfectly periodic, but with typical time scale of 10 to 100 seconds, of fluctuations of different parts of brain. For instance, if you look at the precuneus, you would see the yellow trace in here. There are other parts of the brain which seem to be, even during untriggered spontaneous rest, synchronized. For instance, the red in here, medial prefrontal cortex, the red line almost perfectly synchronized. There also, you can observe anti-correlations if you look at other parts of the brain. But generally, it seems that these spontaneous fluctuations are synchronized in patterns which have meaning. You can actually detect the full zoo of brain networks, borrowing from Smith at now PNAS 2009. So already this is history. That the same networks, for instance, the visual network that you would observe by giving people a task, showing them picture, showing them black screen, and comparing the activity. You can recover it just by looking at the synchronization of the time series of their spontaneous brain activity during resting state. Even eyes closed, you will still find the visual network. You'll find the motor network, you will find the auditory network. Basically, you can map the connectivity of the human brain by just letting people lay in the scanner. That's fun game. So what can you do with that? Well, you can apply the complex network bet. The bet says that instead of describing this in very detail, you will try to come up with a graph which somehow represents the full dynamic behavior. In this case, it could be the graph of the functional connectivity. You have the brain, you parcellate it somehow, you believe that meaningfully into an atlas, let's say the AL region or some other you like. You have a representative time series for each of the regions. You somehow clean it, of course. This is very noisy and dirty data, but after you've done enough cleaning to be happy, not to be perfect, to be happy, you can turn it into a weighted graph or maybe threshold it, and you have a binarized graph of connections in the brain. And somehow you believe that this graph tells you something about that individual brain, or maybe a brain of that species. That's a bet. Yeah, you have to say it's a bet, but it somehow works. Just an illustrative example, it seems to be a bet the researchers take in many other disciplines, not only in neuroscience. I mean, it's a bet that in neuroscience seems to be a favorite game to play. My favorite slice progression, apologies for those who know it. Apologies for those who know the fact that this is my favorite slice progression. In the 1985, the most common word in abstracts of neuroscience was neuron. It's not very surprising, right? In 1995, it was still the most common word, but genes were the second most common word in abstracts of neuroscience. As you move on, at some point, 2015, it was almost as about networks as it was about neurons. And you can see that it somehow goes together with the rise of MRI and connectivity and small words and some of these words. So a bit of extrapolation in here. For those who are interested in the data, I've actually double-checked the 2025. I have no idea about the 2035. So, but you can do a lot of things with these graphs, and you can construct them in very many ways. So how do we actually choose the right one? I mean, there is no right one. There are many models, but let's try to apply Occam's razor into many decisions we take along this route. So I've thrown in term that you probably all know pretty well, functional connectivity, which is defined as the statistical dependence between the activity of some brain regions. But how do you measure connectivity or statistical dependence? Well, if the system was boring like this, you could just use correlation that's shown in here. And basically, Pearson's linear correlation that you learn in your first lesson of statistics tells you all there is to know about the strength of dependence between two variables. If the brain was exotic, like this, or even more, like chaotic system with weird attractors, strange attractors, then, if you compute correlation, you might know nothing about it. Correlation tells you zero, actually, in all of these cases, but there is clear dependence between the variables. So precisely because we believe that the brain is actually a very complex system with nonlinear, maybe even chaotic behavior, almost surely chaotic behavior, we don't want to use correlation, right? Many researchers use many different measures, and there's actually one ultimate way to measure functional connectivity, and that's mutual information. Yeah, that's some information theoretical quantity, which basically says how much of shared entropy is between two variables. And it has nice property. Any type of dependence will be captured by it. Yeah, the other measures have their peculiarity. They're like this or that form. Mutual information captures anything there is. And at the same time, it's zero exactly if some two variables are independent. The question is, should we just switch from using our measures to mutual information immediately? Well, there's a price you pay for using this very generic approach, and it's difficult to estimate it from short time series. There's just the error you have in here is larger than if you knew that it's Gaussian. Then you can prove that the Pearson correlation is actually the most efficient way. So is it worth using mutual information? Because we do believe that brain is very exotic. This is a boring slide. I will just skip it. It basically says the data we used for this study are standard. I mean, if you know how data are analyzed in fMRI, you know immediately how standard it is. If you don't know it, the parameters will not tell you anything. And this is the first result slide about nonlinearity in brain. And that's comparing, for an example brain, what is the relation between computing functional connectivity by correlation? So assuming this is just Gaussian cloud, I will just throw a straight line through it. And the quality of fit is my dependence, versus computing the full mutual information between the variables. And each blue dot corresponds to two brain regions, to some pair. So there is like 4,000 pairs of brain regions. The red line is the theoretical prediction of the relation between correlation and mutual information if the data were really boring, pure Gaussian. And as you look at the picture, you can see that actually the prediction is almost perfect. If you're interested in mutual information, you don't need to compute it. You just compute correlation and plug it into a theoretical formula for fMRI. Of course, you could say that it's a good prediction, but not perfect. The mutual information seems to sort of vary around this theoretical prediction. But what we try to persuade you is that this jitter is actually the jitter due to the estimation of something from few hundred time points you have in fMRI. To persuade you, I'll show you a second picture where we simulated data. We took the real data, we computed the correlation matrix, and we simulated data from a Gaussian sampled with this correlation matrix and tried to estimate mutual information on this mock brain, which is boring. And you can see that the jitter is exactly what we observed. It's as well fit. And in fact, I challenge you to find like 7 differences. Okay, you may find these 3 points. There's a bit more nonlinearity in these 3 pairs of brain regions in real data than in the surrogate. If you even trust me that this is the data and this is the surrogate. Yeah? I think it is. I forgot over the years. This is history, 2011. That was one subject. Just if you play across more subjects, if you're interested, just read the QR code. It seems that the story in here in fMRI is that the nonlinear information is like around 5% of what you can see. And actually this 5% of nonlinearity includes all non-stationarities, artifacts, and stuff that we did not manage to clean, but they do not necessarily say something about the brain. Yeah, it's also a scanning procedure which could create weird trends. You could say, okay, that's what you see for each pair. But what if the graph of the brain, if you use mutual information, is different than the graph of the brain if you use correlation? Each particular link might be almost perfectly captured, but what about the graph properties globally? Well, that's a tough question to answer because it takes a lot of computing time. But we try to go through the procedure of actually generating these mock and real Gaussian brains, comparing them with the real brains in terms of not only each pair, but all the graph properties we could see. No, all the graph properties we were not sort of lazy to compute. And whatever we did, the data, the black line, was always within the 95 percentile of the confidence interval of the surrogates, of the Gaussian surrogate. So it doesn't matter, even for the graph properties. This is not a negative story. This is a positive story. If you're interested in graph properties, feel free to just plug in the correlation matrix into your computation in fMRI. You don't need to go beyond that. I threw in a small story about small word. I've actually checked. Small word is one of the keywords which was trending. And as Cornelius mentioned, it was trending, but it doesn't seem to be anymore. And maybe part of the reason is that, okay, I don't need to show this. You can indeed see small word properties in brain, especially if you take the data, time series, correlation matrix, threshold it, compute small clustering and path length, and also compute clustering and path length of a random graph of the same density. You can compute a single number which tells you how small word something is, so-called small word coefficient. Not everybody likes this coefficient, but it was proposed by Humphreys, I believe. And it's just a ratio of the relative clustering with respect to random network and R-randy type and a relative path length. If this number is more than 1, this means that this is more small word than an R-randy graph. And the brain seems to have this property. However, if you go about it, even a system like this, so vector autoregressive process with completely random coupling matrix A. So if I took your brain, looked at the connections in there, took them out, randomized them, plugged them back in at random positions, and simulated a bit of that, the time series generated from this would have a correlation matrix, which also has a small word property. So just observation of small word in a correlation matrix of anything, it doesn't even need to be brain, is not very interesting per se. Differences between groups is something else. It could be a useful biomarker. But seeing more than in a Shreni is not surprising. If you're interested in more detail about this, again, feel free to have a look at the paper. I will skip this. But if you're interested in topological data analysis, that's another, not graph theory, but also somehow topological approach, which can tell you some interesting stuff about brain. I will just mention one take-home message which you learned along the way. But bear in mind, this is still computed from correlation matrix. It seems that the standard approaches of topological data analysis are able to detect epileptic seizure, but not schizophrenic brain from a healthy brain. As if this was not a topological property, although I can take the matrix and plug it into machine learning, and it will find a difference. I can even do some old school statistical testing, and I will see a difference between the connectivity matrix of a patient with schizophrenia and a healthy control. But if I try to use persistent homology from topological data analysis, it doesn't see it because it's more about where the change is than about what sort of change it is. But still using correlation matrix. So is the whole neuroscience, does it boil down to just computing correlation matrix? Well, okay. Hopefully not. There must be something wrong about this. So let's go. What's wrong about it? Well, one thing which is probably wrong about it, that it's a pairwise approximation. It only looks for each pairs and computes these pairwise correlations. But there are systems like the XOR function applied to even like random fair coins, which are able to generate a dependence between three variables, although each pair is independent. Yeah, so I don't know if you know the XOR function, but basically it says, are these two things the same or not? And this is a very exotic system because if you looked at any pair, original two coins, and each of them with the result are independent, but as a triplet, they share one bit of information. It's actually also nicely symmetric. You cannot say who shares the information about whom, but it's hidden in the triplet. So maybe there is something more to see which functional connectivity as a correlation matrix doesn't see, and that's these higher order interactions. This is an active area of research. You may hear more about it later on. And just some results in this area. There are different ways to capture these higher order interactions. One particular tradition is the so-called connected information approach, which tries to use maximum entropy principle and basically says, is the brain or the system that you have at hand does such that its distribution of its states is the maximum entropy distribution given the pairwise marginals, or is it something else? Because then it means that there is some higher order constraints on it. And how much does the entropy decrease if you between the real system and between the constraints of a first, second, third, and so on order? So this gives you sort of a hierarchy of connected informations of different orders that tell you how much is basically given by these pairwise, triplet-wise, and so on connections. We try to play this game with colleagues from University of Calgary, and it seems to us at the moment that the brain is actually so boring as well, like 90% pairwise in this sense. But there's a lot of open questions. So if you're interested in like higher order stuff, I'm happy to discuss more. Okay, Zoom is ready. I hope it was ready all the time. But this is only about like functional connectivity. There is a different approach, and that's looking also beyond what's dependent to what, on what's causal in terms of what. So what is the cause and what is the target? So people have been drawing not only networks with undirected edges of the brain, but also networks where there's an arrow. The information flows from here there, right? Effective connectivity, term coined by Carl Friston in 1994, or maybe even earlier by others, which people forgot about. And the question is actually the representation by such a directed network a good representation for something? Or are you again missing the option of higher order interactions? If you imagine that you would have a causal system with XOR functions, which of the original, if C is an XOR of A and B, which of the A and B is the cause? Because both of them, like each of them, is independent of C. It carries no information. Only together, they're providing some prediction. So to capture this higher order causality, Jakub Kořenek proposed an approach based on a systematic conditioning and looking for the minima across a subset of conditions to be able to basically ascribe particular causal effect not only to individual variables, but actually to combinations, if needed. But as I said, at the moment, it seems fMRI is boring. So why is it that fMRI is so boring? Well, you may think that, well, okay, one speculation we have with fMRI, that whatever I showed before was fMRI at the level of regions. So it was like few cubic centimeters of brain, their firing rates averaged over space and also averaged over time, because it takes 1 or 2 seconds to take this snapshot of the brain in fMRI. Moreover, we don't measure the neuronal firing activity. We measure the vessels dilating and the oxygenation going up. And that's a process which has typical time scale or like 4, 8, 10 seconds. So maybe we're spatially temporal averaging too much, and we're looking at a window, we're looking through a window through which everything is Gaussian. So let's try to go a bit deeper. So first step, we try to go a bit deeper in terms of number of regions, not 100 regions of the AL atlas, but like up to 1,000 regions of similar to these brain atoms and so on. This is actually a Craddock atlas, which allows different resolutions. But even if we go at very small regions, and even if we looked, I'm not showing it, at individual voxels, the relative nonlinearity is in between 2 or 3%. And it actually corresponds roughly to what you see in a shadow data set. Again, this is a mock brain where we sort of try to simulate it. So it's not about how small the scale is, at least spatially. Up to level of millimeters, nothing changes. And that would suggest that practically it makes sense to use, although mutual information is theoretically equivalent correlation, using correlation gives you more power in detecting differences, predicting outcomes, and so on. So the clinical applicability is actually much higher. So maybe we missed something else. It's the spatial scale plus the temporal scale. So let's move to EEG. If we look at EEG, the advantage, now we are sampling at like kilohertz. Of course, the real signal above 50 hertz in scalp EEG, there's really noisy. But we hope to see a bit more. And there is a little bit more of a relative nonlinearity, but again, it's like 2, 3, 4%, not much to see in nonlinear measures in EEG. Okay, this is very controversial. That's probably why it's not yet accepted, and it's still at the level of preprint. But that's what we see. If we go even deeper, intracortical EEG, so electrodes stuck in the brain. So now they're not only high temporal resolution, but high spatial resolution. They're like millimeter or sub-millimeter contact. So they do not, still their average across many neurons, tens of thousands, but it's not millions anymore. And the relative nonlinearity is now 5 to 10% maybe. To give you a visual idea, this is a Gaussian cloud with a relative nonlinearity of 2%. You just don't know it's not Gaussian. This is relative nonlinearity of 90%. Yeah, you clearly see that this is not Gaussian. Yeah, so for those who are online, this is the 90%. We're nowhere close to this. We're somewhere here with EEG. Like visually, it still seems boring and Gaussian. You really need to go into something like epileptic seizure, high amplitude, highly synchronized activity, to see the nonlinearity of this type, and for the nonlinear measures to give you different answers than the linear ones. Or you go to single neuron data. And we used some publicly available single neuron data. It was still a lot of pain to sort of crawl through the structure, the data structures and what is what, to actually observe nonlinearity. So at the moment, to us, it seems, of course, brain is not boring. The brain is super complex and interesting and chaotic and nonlinear. But at the spatial scale of the neurons, as you look at larger and larger chunks from distance, actually the linear approximation is pretty good, which is a good message for the clinical applications. And I guess for the sake of time and lunch, I'll jump over these bonus slides. Or just Nicola. If you're interested in microstates, you should talk to Nicola. And this is my slightly psychedelic summary. So what I tried to persuade you is that fMRI functional connectivity is purely linear, which is a good thing because it's simple to compute. Please feel free to read the full paper. Topological data analysis can show some interesting global properties of connectivity, such as that the brain seems to be like looking at brain data time series seems to be as sampling hyperbolic space. I don't know why, but that it is the case, you can see it here. Schizophrenia seems to be more about topography than topology compared to, for instance, epilepsy. Quantifying hardware-oriented reductions is a tricky thing. There is some approaches at the moment. It seems that there is not so much of like genuine, not captured by correlation matrix, higher order interactions. But there are some interesting multivariate patterns that you can capture by other ways. Generalizing causality can be done. This is Jakub Kořenek's paper about higher order causality. Accuracy if using correlation instead of MI is higher. That's the important clinical key point. Like, do not use the complex measures if the simple measures captures the same with less noise. Mapping nonlinearity across the different scales. That's the Julius still yet only archive paper, where we went really from fMRI to voxels to EEG, intracortical EEG, and single neurons to finally find something nonlinear in the brain. Yeah, not because you think, but because you see that it's nonlinear. And these are the two things which I jumped over. So if you want the bonus, you need to scan the two last QR codes. With that, I'd like to thank all my collaborators involved in our group or abroad. And thank you for your attention, especially if you're open to collaboration, open positions, but maybe open to discussions. Thank you.